Before we get started today, I do want to remind folks that next month's webinar is going to be on native plants for pollinators and specifically talking about the role that native cultivars might play in plant-pollinator interactions. Make sure to save the date for that webinar if you're interested. Once again, you can register for all webinars on the Native Plant Connection website, which I will grab that link and drop it in the chat as soon as I stop sharing my slides. I feel really grateful to have the expertise of Dr. Meghan Avolio, who is an assistant professor at Johns Hopkins University in Baltimore, Maryland, joining us today to provide an overview of the importance and benefits of preserving genetic diversity in native nursery plants. Dr. Avolio received her Oh, Meghan, I forgot to print out your bio. I know she received her master's degree at Fordham University because we overlapped to some degree at Fordham. And then you went on to complete your PhD at Yale?
Correct.
Is that correct? And then you also had a prestigious fellowship in between those two. I remember that as well.
Yes, I had a Fulbright to Germany for a year, which is really fantastic.
Thank you. A Fulbright to Germany. I apologize, Meghan, for being unprepared in this regard. In no way is it a reflection of, I just hold you in such high regard. Thank you for being here today. For folks who have been on previous Native Plant Connection webinars, we do start the webinar with the land acknowledgement, and that is in deference to the fact that native plants are so closely tied to place. And so we do want to acknowledge the places represented by people who are presenting the webinars. For the past two webinars, I've given a land acknowledgement for where I am in Corvallis, Oregon, but today I want to acknowledge where Dr. Avolio is joining us from in Baltimore, Oregon, and start with the land acknowledgement from Johns Hopkins University. We humbly acknowledge that Johns Hopkins University is located on the traditional and contemporary homelands of Indigenous peoples. Our campus resides on unceded lands of the Piscataway and Susquehannock peoples. We recognize the enduring presence of more than 7,000 Indigenous peoples in Baltimore City, including the Piscataway, Lumbee, and Eastern Band of Cherokee community members. As we gather from places across the country and globe, we honor and recognize Indigenous people and our homelands. Together, we acknowledge the history of genocide and ongoing systemic inequities while respecting treaties made on this territory as a step towards reconciliation and strengthening relationships with Indigenous peoples. We give thanks to the past, present, and future stewards of this land and respect all tribal nation sovereignty and right to self-determination. We aim to hold ourselves and the university community accountable to tribal nations. And then I also like to share one or two plants of cultural importance to local tribes. In the Piscataway homelands, this includes cattails, several typha species, where the young stems of cattails can be prepared much the way that asparagus spears are prepared, and the corms of the cattails are also prepared and can be edible, ground into flour, etc. There's also the white water lily, where the young blossoms are edible as well as the tubers of this aquatic marshland plant. And with that, I will hand it over to Dr. Avolio. Thank you so much for teaching us today.
Wonderful. Well, hi, everybody. I'm Meghan. I'm so excited to be here today talking with you about genetic diversity in plants. As Gail mentioned, I'm currently a professor at Johns Hopkins. I've gotten really interested into genetics of urban plants and planted plants and bred plants when I started working in cities in Salt Lake City. I'm fascinated by weeds. I saw the same weeds in Salt Lake City where I lived, as I saw in my husband's hometown in Turkey. Izmit, Turkey. And so I started to get fascinated in how are weeds evolving to these different conditions in these really different places around the world. And then I also did a lot of work in yards, and I started to get fascinated about the plants in yards. And I would go and see people had lots of poplar trees, trembling aspen trees growing in their yards, which are native to the area. And then I'd go to Home Depot and I'd see hundreds of these trees being offered at Home Depot. And it got me really starting to think about, what are the breeding practices? What does it mean to be native? How does all this play out? And so today my hope is to give you a broad background on DNA and genes and diversity and things underlying diversity, and then ultimately why this matters and what we understand about diversity of trees today. So with that, let's get started. So what is genetic diversity? Genetic diversity is variation in DNA among individuals within a species. So really, when we're thinking about genetic diversity, we're thinking at it within an individual species. And again, it's variation based on DNA sequences. So let's all hark back to thinking about what genetic diversity is, and thinking about both. There's two ways to think about it. We could think about this in terms of how many genotypes are present. And with animals that are absolutely separate individuals, it's really easy to conceptualize this. Like each individual person is a genotype. It's a little bit more difficult in some plant species where they can be connected below ground in the same clones, but right now let's be thinking about in terms of people. And so I really like this figure here. This is a picture of me and my family, and a picture of me and my lab group that I did my PhD with in 2009. And so both of them, there's 4 people here. So there's the same amount of number of genotypes, but how genetic diversity really takes into account how are we related to one another. And so I'm closely related to my family, and I'm not at all related to the women I did my PhD with. And so here we have the same number of genotypes, 4 and 4, but here we would have lower genetic diversity, and here we would have higher genetic diversity. So now I want to harp back to intro bio and really think about, why do we even care about genetic diversity? What is it, and what does DNA give rise to? And so the central dogma in biology is that DNA makes RNA, makes proteins. And if this is all familiar to you, my apologies. I'm just going to try and make sure we're all on the same page. And if this is less familiar to you, I hope I can inspire you the amazing beauty of the natural world and how all this works. So DNA is just a sequence of acids, and together they make genes, certain genes. These genes are transcribed into RNA, and then RNA is ultimately translated into proteins. Proteins is what we observe. So when we observe individual plants, or we observe animals, or whatever we're looking at, we're observing an expression of the DNA, but we're not observing the DNA themselves. And all this gives rise to something that we typically call a phenotype. So a genotype gives rise to a phenotype. And a genotype, again, it's just the sequence of DNA. So what's the sequence of A, G, C's, and T's in each individual person or each individual plant? And then the expression of that is the phenotype, is what we observe. And I'd like to point out that what I like to always say is, like, you can't see someone's genotype, right? Like, no one can look at someone or something and individually know what their DNA sequence is. You need to use a lot of different molecular methods to do that. But you can easily see someone's phenotype. And it's often the phenotype that we're interested in, because that's what we observe. But we have to get that phenotype, we have to understand the underlying genotype. So DNA is inside individual cells, and DNA is coiled into chromosomes. And chromosomes are part of the genetic. When we think about plants a lot, we think about ploidy and how many chromosomes they have. And so here's just an example of how DNA, this double helix of DNA, is wrapped around all these histones, and then these histones aggregate into different levels, and eventually everything turns into chromosomes. And here's an example of stained chromosomes from, I don't even know, I've got this from the internet, from some kind of species. But this is generally what they look like. And when I learned this fun fact when I was in college, like 20 years ago, like really stayed with me. It stayed with me even to today. And so histones, these fundamental thing that DNA has to wrap around to form chromosomes, are basically fully conserved between cows and pea plants. This just goes to show how fundamental chromosomes are and the ability to form chromosomes to the propagation and reproduction of individual species. So if you have a mutation in your histone, you're just going to basically die. And this is why they are so conserved and so similar between cows and peas, which I think is a super fun fact. So as the DNA is packaged into chromosomes, and the number of chromosomes individuals have can determine ploidy and aspects of phenotype. So most animals are diploid and have 2 copies of each chromosome. Some are haploid. And so here in this picture here, each bar represents a chromosome. And here we have diploid, there's 2 copies of every chromosome. There can be triploid, 3 copies. There can be tetraploid, 4 copies. As I said, most animals are diploid, but plants are crazy and can do anything. And so I work a lot on Andropogon giardii, which is big blue stem. And big blue stem can have 6, 7, 8, or 9 copies of its genome. So it's a polyploid, and the degree of ploidy really varies among individuals. So genes themselves are found in chromosomes. And so here is a picture, a diagram of a chromosome. And a gene is just a single segment of DNA that encodes for a specific protein. And so this example here, we're encoding a protein for the color of a flower. A locus is a specific location in a given chromosome. It's like, so you wanted to, for some organisms, the whole genome has been mapped. And you can almost like refer to a page in a book, like it's on page 34. You could say it's on chromosome 1 at location, I don't know, 538. And then someone could go to that area and find this locus where this gene is located. And genes typically have alleles or different varieties of that gene. And so often alleles are coded in biology as different letters, either big A or little a. And so here in this example, we could see that here's the chromosome. I have at the same locus on both chromosomes is the allele for flower color. But these 2 chromosomes have different alleles. We have one that is going to give a purple flower and one that is going to give a white flower. And so I want to take this example just a little bit further to start thinking about how we get phenotypes based on having different alleles at chromosome locations. And so here's the same example where I have 2 parent phenotypes that have purple flowers. But I could genotype them and understand what pairs of their chromosomes have. And in this example, we're working with a diploid plant. It only has 2 copies of each chromosome. And so here, parent 1 has big P for purple and little p for white. And parent 2 has, again, big P for purple and little p for white. And if we were to interbreed these 2 individuals, these 2 parents, we would get if both, not siblings, what's the word I'm looking for? Offspring, get the large P allele, they will have a purple flower. If they get the large P and the little p, if they have the allele for purple and white, they will still appear purple because this is the dominant trait. It's going to override the other trait. And the same for this other parent. But if the offspring gets the little p, the allele for the white flowers from both of its parents, then this plant will have white flowers. And again, this is an example of a dominant allele. Not all alleles are dominant. Not all genes have dominance. Sometimes, like the color of the flower would be maybe a more pink instead of a white or a purple trade-off. But this is just all examples of how variation in DNA gets translated through an RNA into proteins, which gives variation at the phenotypic level that we can observe. So again, what is genetic diversity? Genetic diversity is variation among individuals within a species. And so I hope now that we all are on a shared background understanding of what gives rise to this variation and what gives how DNA is involved in this, and we're going to now go forward and try and understand why should we care about this? Why should we care about having variation, basically in AGCTs inside individual genomes? Well, genetic diversity is the basis for species to adapt to new conditions. And you can imagine a scenario where, climate is changing, where in a genetically impoverished population, where there's not a lot of diversity within this population, there's a low probability of having a certain allele that will be better able to withstand the novel conditions. And so this would put the population at greater risk. In converse, if we had a population that had really high genetic diversity, there's a greater probability that an allele or some variation exists within this population where one individual will be better able to withstand the new climatic conditions. And then this would ultimately be selected for. And so this is one of the reasons why genetic diversity is so important. It's almost, genetic diversity is the basis for which all future adaptation is built upon. And so preserving genetic diversity is help preserving options for species to deal with new climate scenarios and new environmental conditions, such as pests and diseases. Another reason to care about genetic diversity is that there's lots of evidence that more diverse plant populations are associated with more diverse pollinator and insect populations. So this is work that was done on Solidago species. And here on the x-axis is the number of plant genotypes. So I either have 12 plants of all the exact same genetic background, I have 12 plants of 3 different genetic backgrounds, I have 12 plants of 2 genetic backgrounds, or I have 12 plants and every single plant is a different genetic background. And so the first thing that they show is that when you have plots with an admixture of different genotypes, you overall have more biomass in those plots. There's more plant growth when there's more different genotypes of plants or more genetic diversity. They also found in this example that there were a higher number of herbivore insects on these plants, and then a higher number of predator insects on these plants. So the genetic diversity of plant species can populate upwards to be able to support higher levels of genetic diversity at other trophic levels, such as insects that are associating with plants. So how is genetic diversity actually generated? There's 2 ways that genetic diversity is generated. The first is through sexual reproduction and recombination, and the second is through mutations. And so in this example here, and I took this from an online forum, we have the 2 parents, and when they breed together, we have like this blue and this red make purple, this blue and this yellow make green. So there's different levels of diversity within the offspring than the 2 parents alone, or sorry, different levels in the DNA, right? The second is that just mutations can randomly and spontaneously occur through time. And so here we have, again, our same little rhinoceros, where through time, maybe this red allele suddenly has a new mutation and it's a yellow allele, and maybe this blue allele has a red allele now mutates and it's a blue allele. And I'm going to talk a little bit more now about these 2 ways of how genetic diversity is generated. So through sexual reproduction, I'm not going to go into the backgrounds of meiosis and mitosis, but basically there's an example where we all have new combinations of chromosomes from our parents in us. And these new combinations of chromosomes will result in new combinations of alleles, and these new combinations of alleles will result in different phenotypes. And so this is in a way where here the chromosomes of the parents are in all these new possible pairings, and this is resulting in different genetic diversity within the population. A second thing that was really cool that can happen during sexual reproduction is crossing over. And so here's the case where here are the 2 parent genotypes. They line up at one point during meiosis. And when this happens, there's often what occurs is that chromosomes will recombine. So there'll be an entirely new combination of a chromosome inside individuals. And so this is shown here. Here we have the we're looking at 2 loci and different alleles at these loci. So at loci A, we have big A and little a, little a. At loci B, we have a different gene and it has alleles lower B and alleles big B. When there's crossovering occur, now we have a new combination of these loci and these alleles and genes on the chromosomes. And this is another way that the genetic variation is generated. Mutation, as I said, is a different way, and these are spontaneous. Mutations occur fairly rarely. It can occur during sexual reproduction, like in this example here, when chromosomes cross over. Sometimes it doesn't, the crossover, there can be mistakes in that crossing over, and parts of a gene or parts of the DNA sequences can be deleted or duplicated. That's one example. And another example is DNA polymerase, which is the enzyme that is replicating our DNA, makes mistakes sometimes. And so these errors can result in point mutations. It's really excellent. It very rarely makes mistakes, but when it does, this can result in mutations. And so this is just an example in this picture here of what these mutations would look like. And so we can think of each row, 1, 2, 3, 4, 5, 6. We have 6 rows. We can think of this as 6 individuals within the same species. And what biologists will do is line up these DNA sequences to compare them. And so we can see that individuals 2 and 4 have an A where every other individual has a G, and have a T where every other individual has a C. So that's one source of a point mutation. We can also see that individuals 1, 2, and 3 have this region deleted, while individuals 4, 5, and 6 actually have that region still present. And so these are examples of mutations. And these mutations, when they occur if they occur in the correct place in an allele, and if they're not so deleterious that the individual will die, right? Like, let's think back to that histone example. If there is some kind of deletion or substitution in those histones, the individual will probably just immediately die. It would be propagated on. And so this mutation is a big generator of novel phenotypes, and humans have tried to manipulate this to create novel plant varieties. And so some ways that people have tried to get novel plant varieties is through using X-rays or gamma radiation. Here's a figure here of a farmer taking an X-ray machine into the field and exposing corn to X-rays in the hopes that new spontaneous mutations would occur. And here's an example of a gamma field radiation field in Brookhaven National Labs in New York, where they exposed plants to gamma radiation for varying periods of times and then collected the seeds from those plants. And then they sold the seeds from these plants to local gardeners to say, when you grow this plant, do you see anything new pop up? Is there anything interesting? Were there anything new? Were there any interesting point mutations? Now, I read about all this in a book called Evolution Made to Order, which is a fantastic book. And reading about people buying atomic radiated seeds and planting them in their gardens, like, truly made me horrified. I'm glad I didn't live in the 1950s. But this was very popular then, and this is trying to get point mutation variations. Another way to induce mutations in plants is to use chemicals, and colchicine has been used to induce polyploidy in plants. Polyploidy can also be induced with heat or plant injury. But when you increase polyploidy, and here we're going from maybe being a diploid to a tetraploid or a diploid to a polyploid, simply just having a different number of chromosomes can result in different phenotypes. Generally, tetraploids are bigger plants than diploids. There's a larger genome. This results in bigger plants. These are just some examples from hops. You can see here, the B is the tetraploid, is a bigger overall than the diploid. And this has been used extensively as well. This was also popularized as something people should be able to do at their home and try and induce new mutations in plants. Thank you for sharing that book, Gail. So these are all, and now I want to talk about what happens with this genetic variation and how this can change through time and how this results in different evolutionary processes. So evolution is just the change in allele frequencies in a population over time. So it's just, are there going to be more purple flowers through time or more white flowers through time, right? And it's just these alleles. And it's all this variation that comes about through variation in DNA. There is generally 5 ways allele frequencies can change through time: natural selection, genetic drift, and artificial selection as well as migration and sexual selection, but I'm not going to be talking about these two today. I'm really going to be focused on natural selection, genetic drift, and artificial selection. And I'm going to try and go over how these processes work and then make linkages to horticultural practices and breeding of native plants. So natural selection is obviously the most famous form of evolution. It was theorized by Darwin in the 1860s and I'm blanking, and Alfred Wallace. Okay, so you need the fundamental basis of natural selection for it to occur is that you have to have genetic variation to have variation in phenotypes. If every organism is exactly the same, there can be no natural selection because natural selection is based on differential fitness. So if you have a change in an environment, you're going to have a change in the selection pressure. And you need three things for natural selection to occur. You have to have variation in traits. You have to have this variation be heritable. And this variation has to result in differential survival and reproduction. So again, if every individual is the same, if we only have one clone in the exact same genotype everywhere, we can't have natural selection because it's differential fitness of individuals. I'm going to walk us through an example of natural selection with finches in the Galapagos, which is a very famous example. So I'm going to talk about work done by Rosemary and Peter Grant. They studied finches in the Galapagos for over 20 years, and they noticed a lot of variation in the ground finch, in the beak size of the ground finch. And so here's all birds of the same species, but you can see they can have really small beaks to pretty large beaks. And they found that this characteristic is really heritable. So first thing that I said you need to have natural selection is you have to have variation in a trait. And this is the range of beak lengths, beak depths, beak sizes that were observed in the ground finches. And you can see that there's a lot of variation here. This is a histogram. So there were, let's say there's one individual that had a beak depth of 6 millimeters. There was around one individual that had a beak depth of 14 millimeters. And most birds had beak depths between 8 and 11 millimeters. Right, because you can see here on the y-axis, more finches had beak depths of that size. The second thing is I said that this variation had to be heritable, and they did very meticulous work to link the beak depth of the parent and the beak depth in the offspring. And what you see here are strong correlations. So parents that had smaller beaks had offspring with smaller beaks, and parents with bigger beaks had offspring with bigger beaks. So this is really good good evidence that the trait is heritable. And finally, the variation should lead to fitness differences. And so here, birds with bigger beaks are able to feed on a greater variety of food sources. And so birds with large beaks can feed on soft, medium, and hard foods, but birds with smaller beaks could only really feed on foods that are soft. So this we would assume would result in some kind of fitness difference. So what the Grants observed is that they saw a big shift in the beak size distribution of finches following a really large drought. And so this drought occurred in 1976, 1977. And before the drought, the average beak size was 9.5 millimeters. After the beak drought, the average beak size was 10 millimeters. And what occurred during this drought is that the finch population went from having over 1,400 finches and plummeting to having around 200 finches. So this was a very strong selective event, and there was a drastic reduction in the population size. And of the birds that survived, only birds that had the larger beaks survived. So here we see percent survival. Birds with beaks over 10 millimeters had much higher levels of survival, while birds whose beaks were 6 or 7 millimeters simply were not able to live through this drought event. And what occurred was that plants with larger seeds were able to reproduce and set seeds. So the only food source was larger seeds that only birds with larger beaks could break open and ultimately eat. And this is a really nice example of the population had the important existing variation within the population to withstand this novel climatic event. You can imagine if the population only had small beaked birds, this population would have gone locally extinct on this island where this occurred, right? because all these birds died. This is one example why it's so important to have genetic variation within populations. Another way that gene frequencies can change through time, another mode of evolution, is just genetic drift. And genetic drift are random fluctuations due to chance events. Genetic drift is really important for small populations. And so we're thinking about rare populations or species that have been lost a lot of numbers through habitat fragmentation. This can be a much more important process. It leads to genetic drift is just random changes through time. There's nothing driving it. It's not necessarily beneficial. It's not necessarily not beneficial. It's just change through time. And it's really important for small populations. And so we can think of an example here. We're taking gumballs out of a gumball machine. 75% of the gumballs are orange, 25% are green. If I randomly take a small population, like for example, if I have a founder population, I can now have only 40% orange and 60% green. So this is a big change in allele frequency. It's also totally random. And now this new population is going to be very different than the founder population. This is something called the founder effect, where the genetic diversity of the founding individuals sets the trajectory for that local population. So here's an example of genetic drift within prairie chickens. And so prairie chickens, as in their name, prairies, they like prairie habitat. And so the prairie habitat has basically been expatriated out of Illinois. We see that prairie covered a large swath of Illinois in the 1820s, and there's just really two small remnant patches in the 1990s. And they compared the number of alleles at genes in the 1930s to the 1990s. And they found that in the really fragmented Illinois, where so much habitat was lost and where the populations are so much smaller, at 6 genes, where there used to be 31 alleles, there's now only 22 alleles. So we saw a drastic loss in genetic diversity. And this loss in genetic diversity, this loss in the number of alleles, also has fitness effects, where 93% of eggs that hatched survived in 1933, less than 50% of eggs that hatched survived in 1950. And so again, this is just, these alleles are being lost in the population through the drastic winnowing of the population. And then for whatever reason, like one of these birds could randomly die with a unique allele. And once it dies, if it doesn't reproduce, it'll just be lost from the population. But it didn't die for any reason associated with natural selection. Let's just say it, like, I don't know, was hit by a car or something, or it was just taken away randomly. It had nothing to do with its actual fitness. And this is what genetic drift is. And I think genetic drift is really important for thinking about breeding practices. And finally, I want to talk about artificial selection. When Darwin wrote his book on the origin of species, he discussed artificial selection. He spoke a lot about dogs. Artificial selection is when we as humans breed species for traits that we really find desirable. And so corn is a wonderful example of artificial selection. Here is the ancestor, where we have both the male and female parts on the same flower. And then in modern corn, we have the male parts and the female parts entirely separate, and the female parts that are the delicious edible part that we eat are much greatly enhanced and made bigger through artificial selection. A lot of the food that we eat undergoes artificial selection. Okay, so natural selection should lead to local adaptation. So in this cartoon example here, I have the exact same species of a plant, but I have one ecotype that's at high elevation and one ecotype that's at low elevation. And these are what we would call two different ecotypes. So local adaptation is when individuals are well adapted to their local environment. They have higher fitness at this local environment. So if I were to take an individual from the high elevation and grow it down at low elevation, it would not grow as well as if it was at high elevation. And same thing vice versa. If I was to take a plant from the low elevation and try and grow it at high elevation, it would not grow as well as it would if it was growing in low elevation. These are examples of local adaptation. And so again, as I said, this would be a low elevation ecotype and a high elevation ecotype. Ecotypes are genetically distinct populations. So we would be able to detect these differences not only phenotypically, but on a genetic level. And they're locally adapted to their environment. And so I want to spend a little bit more time thinking about local adaptation. Here's examples of local adaptation within an Achillea species going across the Sierra Nevadas into the Great Basins. And plants, what we're seeing is great variation in plant height and plant size. And so here is just a histogram of plant height. So there's lots of variation in plant height. You can see this is a big bar at the westernmost source of the range, but on average, plants are about, oh, I don't know what this is. Let's guess like 90 centimeters tall. And then there's a lot less variation right on the edge of the mountain range of the Sierra Nevadas here. And the plants are a lot shorter. Maybe plants are about 20 centimeters tall. And so all of these would be evidence of local adaptations to very different environmental types found across the Great Basin. Scientists really like to try and study and understand local adaptation. And to do that, we like to grow plants. We like to either do reciprocal transplants, like taking plants from one location and growing them in another location, or grow them just all together in a common place like a greenhouse. And so here's an example of local adaptation with Arabidopsis thalia. This is a model organism in plants. It's very studied to understand what DNA sequences give rise to what phenotypes. And so this is an example where they collected plants in Norway, Sweden, and Germany. And I just want to focus on the results from Norway and Germany. So here they found that plants that were from Norway had higher survival than plants from Germany when grown in Norway. So here the plants are grown in Norway. This is through time, and this is the percent of plants that survive. And you can see that plants from Norway, grown in Norway, have really high survivalship through time. But plants from Germany grown in Norway have really low survivalship through time, where less than 40% are surviving after living in Norway for 3 years. They did the same thing, but in Germany. So here they have plants being grown in Germany, and they have plants from Germany had lower survivorship, but higher survivorship than those from Norway. So about after 1 year, about 60% of plants that were from Germany survived, but only about 30% of plants from Norway survived when being grown in Germany. This is really great evidence of local adaptation, where the German plants are locally locally adapted to conditions in Germany, and the Norway plants are locally adapted to conditions in Norway. Another example of this is work done by my graduate student, Eric Yee, looking at hairy crabgrass. Hairy crabgrass is a grass that's in the family Poaceae. It was introduced to the US in the 1700s, and it's a pretty noxious weed, but because it's non-native and was introduced in the 1700s across the US, it's had the same amount of time to basically locally adapt and evolve in very different cities. So we did a comparison of plants growing from Minneapolis and plants growing from Baltimore. As you know, Minneapolis is a lot colder than Baltimore, but they have fairly similar precipitation. So we collected seeds and we brought them all back to our greenhouse and grew them for a summer. And what we found is that plants from St. Paul, Minneapolis, St. Paul flowered much earlier than did plants from Baltimore. So on average, plants from Minneapolis flowered after 40 days, but plants from Baltimore flowered after 50 days. This makes a lot of sense about local adaptation. There's a shorter growing season in Minneapolis. Plants are going to want to reproduce more quickly, set their seed more quickly before frost comes again, and then they would be killed for the season. And in the growing season is longer in Baltimore, so plants can take more time to acquire more carbon to potentially set a larger number of seeds. So these are both examples of plants being locally adapted to their conditions. When thinking about ecotypes, there's lots of benefits to using local ecotypes. I've taken a lot of this information from Extension Outreach from University of Maryland. It's thought that local ecotypes are better able to grow in local soil and climate conditions, right? They've evolved to these soil conditions, they've evolved to this climate condition through natural selection, so they're going to be better able to persist. There would also be a better resource for pollinators. Local pollinators have evolved to withstand or potentially like better the pollen sources created by these local plants. And they might even be more resistant to local herbivores. Here they've evolved as well to what plants would be eating them. So they have a lot of advantages to succeed in these local environments due to natural selection. So I want to end today really thinking about how breeding of plants can affect genetic diversity of populations. And so there's some practices that would reduce the genetic diversity of plant populations. So if we're breeding plants through sexual reproduction methods, if you only have a few parents that you're breeding, this would result in a genetic bottleneck, right? This would potentially be very similar to a founder effect, where we just have lower diversity overall. And then I have 3 pictures here of actual asexual reproduction. So lots of plants reproduce asexually on their own. It's one of the ways that they reproduce. And so here's an example of a plant tillering, but we could also have examples of plants suckering. And this is just natural asexual reproduction that can be used to clone plants. And humans can also take cuttings, they can graft, and they can do tissue culture to propagate plants with genotypes that are desirable. And depending on how native plants are propagated or collected could inform on, how native are they exactly. And so when we're thinking about native plants, a very good native would be locally adapted to the area. It's undergone natural selection, and sometimes this would be called a wild type. And I'm going to give an example here with Echinacea purpurea. Here's the Echinacea wild type, and here are 3 different cultivars of Echinacea. A variety is an example of a plant that's been found in the wild. It has some unique mutation that gives it a trait that we're really interested in. So this could be potentially something like having white petals, right? So there's this natural variation in the field. This genotype gives rise to a different phenotype. And typically varieties are propagated through sexual means. And so these plants have undergone natural selection, right? They're locally adapted, but they have this extra special interesting trait we're interested in. But they've also undergone artificial selection because now we're actively breeding them because they have this desirable and interesting trait. And finally, there's cultivars. These are plants that are typically hybridized or bred for specific traits, typically through asexual means. Cultivars will have the lowest genetic diversity among them. You can imagine if you are taking, go up here, if you're taking the same plant and cutting it 3 times and then planting these out in a field or in a yard, they're going to have the exact same genotype among them. There is no genetic variation within them. And so cultivars will have the least amount of genetic diversity within them. And I certainly don't have this answer, but I think it's an interesting intellectual thought exercise, like, are cultivars native? What does it mean to be native if this has been bred in a lab and is not very locally adapted? And finally, I'm going to end with some examples of genetic diversity within trees. So I am very interested in how plant breeding practices are affecting the genetic diversity of urban tree populations. And there's very little work on this, but the work that has been done suggests that there is a genetic diversity problem. So there's an example of tilia, which are lime trees being sold at nurseries. There's very little genetic variation among trees that are being sold at nurseries. Generally, there's just 2 cultivars in Europe. There's an amazing example of the exact same genotype of a European lime has been cultivated for the past 200 years, which means the same different people, because people don't live 200 years, have been going back to the same tree, which has lived 200 years, and taking cuttings or doing graftings and selling this. And so this means that across space, there's really no genetic diversity, right? If all these exact same cuttings are being planted, but also through time, there's really been no diversity. And finally, they found that another study compared lime trees that are being grown across Europe, and they found that mostly they're just two clones are really being grown and bred. So that would suggest that there's very little genetic diversity among the urban populations of the species. Here in the US, there's been a little bit work done on the London plane tree. And this is work where they found that there was much greater variation of clones being offered about 50 years ago than that's being offered today. So which suggests that a certain number of cultivars are now really of interest, are being very strongly propagated and sold, and it should be resulting in a loss of genetic diversity within this species. So that is all I have prepared for you today. I just want to end with a summary. Genetic diversity is the variation among individuals within a species, and it's the basis for adaptation. And it's generally generated via mutations and sexual reproduction. Evolution is the change of alleles over time, and natural selection will result in a species being locally adapted to conditions, both in terms of who's eating it, who's pollinating it and what are the soil and climate conditions. And artificial selection will result in species having traits that humans desire, not necessarily that are well adapted to local conditions. And finally, local adaptation have ecotypes that are genetically distinct and are locally adapted to their environmental conditions. That's all I have prepared. With that, I'm very happy to take questions.
Thank you, Megan. That was fantastic. I've learned so much as an entomologist who needed a good refresher on basic genetics and plant genetics in general. A question from Mara, wondering, is breeding across ecotypes advantageous for increasing genetic diversity?
That's a great question. Yes, it would definitely increase genetic diversity. You would imagine that each local type, ecotype, as they're really evolved to this one condition, would have very different genetic backgrounds. And so that would increase genetic diversity, but that would then result in them not being locally adapted to probably either location. So there's definitely trade-offs in what's happening there.
Thanks, Megan. I had a couple of questions.
Yeah.
And it's trying to show how little I understand about plant genetics, because it just is crazy to me to think about that plants can have 7 sets of chromosomes. I don't even, like, that blows my mind. I'm just wondering.
I know, plants are crazy.
Plants are crazy. I'm just wondering if you have any knowledge of what ploidy level, like having a fewer versus more numbers of chromosomes, might mean in terms of impacts of artificial selection. Does that mean that you go down to a genetic bottleneck quickly when you're selecting plants that have low ploidy level versus high ploidy level? Does it mean that plants that have high ploidy level are naturally going to have more of the raw material for adaptation to environmental change just by having more chromosomes? I don't know.
So I will say, a lot of people who do genetics like sexually reproducing diploid organisms. They want animals, right? And so a lot of the whole basis of understanding adaptation and genetics are based not on asexually reproducing polyploid plants. And so a lot of your questions, Gail, it's still not very well understood. But I think there's certainly, being polyploid doesn't necessarily mean you have more genetic variation. You can have 7 copies of chromosomes, but each chromosome could have the exact same allele in it, right? There's maybe a greater probability that there's variation there, but it doesn't necessarily mean that there's going to be. And I think understanding about how this plays out, in terms of adaptation and like with artificial selection, I don't have a good answer for you. I do know that some genotypes or some chromosome levels cannot sexually reproduce. So like if you're Andropogon giardiae and you have 7 or 8 copies of the genome, you cannot sexually reproduce. You can only reproduce asexually. And then you can start to think about, well, what's the species then? And is this an evolutionary dead end? And these are all really open, great questions. I don't have answers.
Yeah, thank you. As an entomologist, I've always been focused on the diploid view. The plants just threw me for a loop. There's a question as how many generations should plants be bred to make them better suited to local conditions?
That's a fantastic question. That's going to depend on so many factors. So one factor is, you can imagine if I'm taking a plant from Alaska and I want to start growing it in Florida, these are extremely different environments, right? That's going to result in really, really strong selection, right? Because it's such a different environment. Most individuals aren't going to be able to grow and reproduce. So you're probably going to need fewer generations to make them locally adapted because it's such a strong selective bottleneck. But if I was to take a plant from Baltimore and try and grow it in Florida, they're really probably fairly similar environments and would probably take a lot longer for them to be locally adapted, because many of the plants from Baltimore are going to be able to grow fine and reproduce fine. And so I think it depends on a lot of things. I think for most examples of local adaptation occur on the level of like research is changing how people think about this, but a lot of people think that evolution would not happen on human timescales. So like it would be a moot point. It would be really hard to actually do this. There is evolution, there is more examples of evolution happening on really short timescales, but I don't have a good answer for how many generations. I think it would depend on the plant and really how different the conditions are.
Thanks, Megan. Another question, so appreciation for the great review of genetic diversity. How does this apply to gardeners? How important is it to choose regionally native plants when choosing native plants for urban landscapes? Is question number 1. And then question number 2 is, how do you feel about the term nativar?
I saw the term nativar while preparing this talk, and I don't have an opinion on the term nativar. It's catchy as a word, but scientifically I don't have an opinion yet. And for regionally native plants, I think that's really important. If you want to plant, a native garden that's going to have the most benefits for local species, like if you want to have a pollinator garden, for example, and you want native plants, I think it'd be really important to have locally sourced native plants. So, a lot of species have a range all across the US, right? But if I live in Baltimore, if I wanted plants in my native garden, I would try and source plants from around Maryland. I think would have the most beneficial effects.
Thanks, Megan. I'll mention that there are folks on our Native Plant Connection team who have very strong opinions on the term nativar. We've debated it within our group colloquially. Hi, Ryan. I'm in the middle of the webinar, sorry. Okay, another note, someone just sharing a Popular Mechanics article about X-raying peanuts to create flowers and plants. Thank you for sharing that note. And then I also have a question kind of like relating genetic adaptation, ecotypes to gardens and gardeners. The environments that a lot of these plants have evolved in or have been subject to natural selection in are fundamentally different than most urban and suburban yards. And so I've always wondered about the value of choosing a locally adapted ecotype when oftentimes the soil conditions and maybe even like the microclimatic conditions, if it's something that evolved in a forest and we're putting it in a sunny suburban or urban yard. Do you have any thoughts on the relative value or importance of placing ecotypes into garden habitats that might be fundamentally different than the location where those plants evolved from?
This is a great point, and this is why I think thinking about what planted plants are doing in yards will never cease to fascinate me and just be wonderful to think about. So I think 2 things. I think for the plant itself, generally we're creating conditions in yards that should be a little bit nicer for the plant, right? We are growing it. Most time, if you're in an arid area, it's going to be watered regularly. Plants are going to be happy with that. Typically, there's some level of fertilization. That's more nutrients for the plants. So I don't think it would be stressful for the plants to be grown in these different, really suburban habitats. And I do think from the thinking about who's associating with the plants, I think it matters at that level. And so I think, if we're plants themselves, I guess, okay, I want to talk about 2 things here. I think it's important for 2 things. I think first, at the genetic level, like if a plant is pollinated or insect pollinated or wind pollinated, there can be breeding between local plants that are not in yards and actually plants in the yards. And so having local ecotypes is going to help preserve that local diversity within the region, which could be really important. And second, if there's insects or birds that are flying around and easily dispersing, or other animals that can easily disperse from these populations that are growing natively and populations in yards, those insects or those other animals will be better able to interact with local ecotypes compared with foreign ecotypes. So I agree from a plant growth point of view. I don't think necessarily the conditions of plants are growing in, it matters as strongly, but I think it matters for those levels of genetic diversity and then who's associating with the plants.
Thanks, Megan. And I have one last question. Oh, one other came in. Megan's last point about pollination between garden plants and wild populations may relate to preserving rare populations.
Yeah, so if you have a local population that is rare and an ecotype, and if that's being planted in yards, then absolutely that would be helping that rare population. If you're planting in cultivars of that same species that don't share the same genetic background, it could potentially be detrimental, right? Because then you're going to have cross interbreeding and gene flow among locally adapted ecotype and a foreign ecotype.
Thanks, Megan. And then my last question is about production practices in plant nurseries, which oftentimes use asexual propagation methods for efficiency of production or to be able to produce at scale. If you are using an asexual propagation method for efficiency of production, like, does it make sense to like stop and try to interject like novel genetic variation into that asexually propagated?
Yeah, I would think so. Like, there's obviously trade-offs, and I fully recognize that if you need to propagate a lot of plants quickly, asexual methods are really wonderful and can enable that. And so I would think one way to do it is maybe every year use a different genotype to propagate asexually, right? Take clippings or cuttings from a different plant. And so just that itself would create diversity through time. Like the example of that European lime tree where it's the exact same genotype over 200 years. I just love that example. It boggles my mind. I think preventing things like that, anything you can do to inject any level of diversity is really helpful.
Thanks, Megan. And one more question came through the Q&A pod. Do you have thoughts about how to choose both 1, species, and 2, parent plants for collecting seeds and propagules when we know the climate is changing and local conditions will be significantly different within the lifetime of the plants?
This is a fantastic question and something that is deeply debated within the scientific community. And I will say that I personally don't know the answer or have a strong opinion, but what a lot of people are doing, and it could be considered advantageous, is collecting populations from more southern locations to then plant them in more northern locations. And so things that are really long-lived like trees, if I plant a seedling now, it'll hopefully be here 50 years from now. And if I plant a seedling from Baltimore in Baltimore, maybe it won't be as able to withstand the climate in Baltimore 50 years from now. But if I plant a seed from South Carolina in Baltimore, maybe 50 years from now, it'll be better adapted to the future conditions of Baltimore. So that is certainly something that can be thought about and people are doing. And it's, yeah. And I do think it's maybe one of the tools in the kit of how to help species be better able to adapt to different climatic conditions. Of course, so this is then all shuffling all genomes, and that would get rid of local ecotypes. So it's a complex thing. I don't have an easy answer.
Yeah. Thank you, Megan, and thank you all of you for showing up today for a complex topic and for advancing our own understanding of the importance of genetic preservation for native plant populations. Remember once again to visit our website, Native Plant Connection, to access recordings, closed captioned of the webinars. Yes, and then also to register for future webinars. Greatly appreciate your time today. See you next month to talk about pollinators, native plants, and native cultivars. See you then. Bye now.